Good morning. My name is Mitchell McAndrew in the lab of Dr. Odinone Lapanet. Today we are pleased to have Dr. Stefan Hill, one of the directors of the Max Planck Institute for Multidisciplinary Sciences in Göttingen and the Max Planck Institute for Medical Research in Heidelberg. Dr. Hill received his doctorate in physics from the University of Heidelberg, training under Dr. Siegfried Hunkler. He then took research positions at the European Molecular Biology Laboratory at the University of Turku, as well as visiting scientist position at the University of Oxford. Dr. Hill joined the Max Planck Institute of Biophysical Chemistry in Göttingen in 1997, where he continues as director. Dr. Hill has received many prestigious awards throughout their career, including the 10th German Innovation Award, the Cavalli Prize, and the Nobel Prize in Chemistry in 2014 for the development of super-resolution fluorescent microscopy, along with Eric Beitzig and William Moriner. Dr. Hill, both theoretically and experimentally, showed that you can improve the resolution of fluorescent microscopy beyond the previously thought limitation of half the wavelength by decoupling the resolution of the microscope from the diffraction, allowing for nanometer scale resolution. This opened up a new field in science of super-resolution fluorescent microscopy, also called fluorescent nanoscopy, which is able to observe molecular structures, track processes occurring within living cells. Today we will hear about MinFlex, a recent super-resolution method that has reached the resolution of a fluorophore molecule, allowing for characterization of dynamic processes and tracking sub-nanometer scale details. Please join me in welcoming Dr. Stefan Hill.
Thank you very much for having me today. I'm going to speak about a very simple problem, namely how to localize a fluorescence emitter in the focal plane of a microscope with maximum accuracy, precision, and speed. Now, localization has been around for many decades, and the way people usually think of localization is to have a wide field illumination of the focal plane of an epifluorescence microscope and then record the fluorescence that is emitted by the single molecule whose position we want to find out by the optics. And then the optics projects the fluorescence onto a camera. Now the camera, consisting of many pixels, produces a diffraction spot of fluorescent light, which is usually called the point spread function of the system. Now the narrative goes as follows. It is easy to find out where the molecule is positioned in space by just calculating the centroid of the diffraction blob, because the position where the maximum is essentially coincides in space with the position of the sample of the molecule in sample space through simple geometrical optics. Now the precision with which we can find out where the molecule is actually located scales inversely with the square root of the number of detected photons n, and the more photons you have, the better the localization precision will be. Now, it's very obvious that this kind of localization, which by the way has been used for decades in many biophysical experiments and of course also in the imaging methods called Palm and Storm, essentially relies on the maximization of the number of fluorescence emissions from the photon. And this, of course, is inherently a drawback because fluorescent molecules, they tend to bleach. Not only they bleach, they also blink, meaning they go off for a while, then they're silent, then the molecule can move away, for example, and they come back, and then, of course, you have a problem. And the problem is that you also need to wait for a long time, because if you need to maximize the number of photons n, then you have to wait. That's inherent in the concept. And there's another problem, which in my view is even more serious and sometimes forgotten. Namely, molecular emission means that the molecule emits like a dipole, meaning emission is not possible in this direction or in that direction. Emission can go only in this direction, like this, orthogonal to that transition dipole. As a result, the pattern of the diffracted light of the point spread function, as we may call it, is shifted to the side because the emission kind of goes to the side. In other words, the actual position that you of the molecule does not perfectly coincide with the maximum of the diffraction pattern. It is shifted, and that can amount to 10 nanometers, 50 nanometers, or even 100 nanometers, particularly if the molecule is off the focal plane, above and below the focal plane. And so all these factors kind of contribute to limitations when it comes to localization. So while the precision may be very high, very, very high, because you have many photons collected it may be that the molecule might be in the wrong place because the accuracy is not correct. And this is why I decided to come up with a totally different way of localizing individual molecules, which does not rely on an epifluorescence illumination, but on a concept that I'm calling coordinate targeting. So what do I mean by this? Now, the idea behind this concept is to target a certain coordinate with a light pattern, namely an illumination pattern. So imagine we have a laser beam, and the laser beam is converted into a donut beam. Now, a hallmark of a donut beam is, of course, the central intensity minimum, which is perfectly defined. If that minimum is produced with a laser beam, is perfectly defined because the minimum is exactly at the point where never ever a photon, an illumination photon, is showing up. If you're slightly out of the minimum, there is always some photon showing up because the laser produces many photons as a source of light, so there's no shortage of photons, and therefore this position here is perfectly well defined and also easily measured. Now, since this position is now defined by the laser, the whole localization process boils down to finding the distance between the unknown position of the molecule and the known position of the zero of the donut beam. Not only we know exactly where the beam is, we can also move it in sample space, so in the focal plane, because we can place here a beam deflector that moves the donut zero around and moves that coordinate position around in sample space. And then, of course, we can use this known position to find the position of the molecule in a very simple way. So what is this simple way? It's very easy in retrospect, at least. Well, the brightness of emission of this molecule will, of course, depend on the relative position of the molecule with respect to the donut zero. Here, for example, there will be some excitation because there is green light, so relatively many green photons arriving, exciting the molecule, and hence producing fluorescent photons here at the detector. Note there is no camera in here, there is just a point light detector. And then, if the two coincide in space, so if we managed, for example, to move the donut zero just on top of the molecule, then of course at the donut zero there won't be any excitation, and as a result no fluorescence emission. So the detector will not measure any count, so the fluorescent molecule will be silent, and then we know exactly where the molecule is, namely at the known position of the donut zero, because the position of the donut zero is always known because it's controlled with the beam deflector and perfectly defined. If it's slightly off, if the donut zero is slightly off from the molecule, then of course the molecule will emit fluorescence light, but much less than before, because here the intensity is lower, and this will be seen by the detector. So in other words, the brightness of the molecule will tell us how far the molecule is away from the donut zero. Now, the big question is now, why is this concept superior in all three aspects with respect to normal localization? So it requires fewer photons, it gives more accurate number, and as a matter of fact, you will see in a moment, it is also much faster. Why is that? In order to explain this in a very simplistic way, and with my last name being Hell, I decided to come up with a demon. And the demon, of course, knows something that we humans don't know. Namely, let's assume for a moment that the molecule starts moving. And the demon has the capability of moving the donut zero within no time, say within an attosecond. And attoseconds have become very fashionable lately. It moves the donut zero within an attosecond on top of the fluorescent molecule. Now, if this is the case, then the molecule cannot emit any photon because the zero is always on top of the molecule. Yet the demon can trace the position of the molecule based on the absence of fluorescence emission. So in other words, we would get here a perfect trace of the molecule, picometer precision if you will, just based on the fact that there is no fluorescence emission at all. And this can happen only when the position of the molecule always coincides with the known position of the donut zero. So in other words, this thought experiment tells us that in principle there's a way to come up with a new type of localization which does not rely on the maximization of the number of fluorescent photons, but just on the opposite. In essence, here in the thought experiment with the demon, it doesn't require any fluorescence emission, so no fluorescent photon at all. And this is kind of mind-boggling, especially if you compare it with the previous case where you had to maximize the photons. And then there's also another interesting aspect about this. If you look into the details, you will find out that there is also no wavelength dependence of the localization precision. And this is kind of mind-boggling, especially for a physicist who is trained that, if you have freely propagating waves, basically any measure that you have with light scales with the wavelengths, must scale with the wavelengths. But this is not the case here. Why is that? Well, in essence, you can argue that the molecule is kind of a point, and also the donut zero, at the very minimum, is a point. And so what we do actually, we kind of match 2 points. And of course, in that case, it doesn't really matter whether the donut is made of green light, yellow light, blue light, ultraviolet light, infrared light, ultrasound, or whatever. So any type of wave, as long as we measure or try to measure the absence of the backscatter light of the fluorescent light, we can have this ultimate precision. It really shows that it's a way to really overcome the limit set by diffraction and the wave nature of light. And then the dipole orientation goes away, largely because, if there is no fluorescence emission, why would the orientation of the molecule and transition dipole of the molecule count? The truth is, the closer this distance gets actually between the zero and the molecule, the lesser the orientation of the dipole dependence comes into place. And so this is, of course, a big advantage over the previous case. However, in reality, there's no such thing as a demon. So we have to do something that works in the real world. And what we do in order to make this concept viable in practice is we replace the demon with a controller, meaning that the controller, of course, counts the number of detections that will emerge as a result of the fact that the position of the donut zero will not always overlap in space with the position of the molecule. So if the molecule starts moving like this, there will be some emission and hence some detection that is counted by the controller. But the controller will instantly find out where the molecule is, so to speak, from the brightness of the fluorescence emission for a certain intensity that is found here close to the donut zero. And so in this way, of course, we can track the position of the molecule with a certain precision that very much, of course, depends on the average distance between the donut zero and the position of the molecule. So the better the match, of course, the better this localization precision is. Importantly, this concept called min-flux now allows us to have a precision, and this is the new thing here, that does not just scale with the square root of n. That is something that comes out of the photon statistic, but has this dependency on the average distance, residual distance between the donut zero and the position of the molecule. And the smaller this distance is, so the better the match between the two, the smaller sigma gets, so the precision value gets, meaning that precision goes up, because smaller sigma means higher precision. So in other words, a min-flux now allows us to get higher precision than normal localization that is largely independent of the wavelengths with the same number of photons, and the trick is make the distance between the two as small as possible. The closer you come to the demon situation, the better you excel at localizing the molecule. Now, that's the concept. The question is, how good are you in practice? And at this stage of development, as a rule of thumb, you can say that we need, for the same localization precision, 100 times fewer photons. So 100 times fewer photons, that's a lot, because it inherently means that you can be 100 times faster if you need the same precision. And 100 times faster means that you can, for example, track a fluorescent molecule in 100 times smaller period of time. And that's why we looked into relatively quick processes that couldn't be observed so far. And to give you a ballpark number, so how quick can we be with min-flux? If everything goes well, we can get a spatiotemporal resolution that is in the range of 1 nanometer per millisecond. An interesting question is now whether we can see the movement of proteins also in a living cell. not just on, say, an artificially prepared microtubules with kinesin coming from solution or something like that. And that is something really interesting because, in that case you inherently have the right ATP concentration, you have endogenous protein levels. And indeed, this experiment was done. Jonas Schleske, who is a PhD student in my lab, he managed actually with a CRISPR-Cas system to endogenous label dynein, and then to track, of course, by attaching a fluorophore on a halo tag, actually the position of that dynein right in the cell, in the living neuron. And this is the first time, to our best of our knowledge, where individual steps were seen in living neurons. And this again shows the strengths of MinFlux. You can zoom in, so to speak, on a protein in a living neuron, because you have a kind of focal detector that suppresses background, so it's very important, and then see the steps. And this couldn't be seen with other methods in this non-invasive manner, or minimally invasive manner. So this is actually the trace. You see here there's various steps that dynein makes, on average 8 nanometer steps, but it's very interesting to see those pauses. And of course there is this notion that the dynein, kinesin, since they're responsible for different directions, retrograde, anterograde transport, that there is a tug of war between them when the cargo has to be sent to the periphery of the cell or back to the nucleus. But we've seen this tug of war relatively rarely, so we anticipated to see more of this tug of war, but we didn't really anticipate to see quick reversals between this direction. And these are shown in here. So within a period of time that is kind of in the ballpark of the step duration, it's possible for dynein, so to speak, to go back and change its direction. And so this is not kind of anticipated. Again, my point here is that MinFlux has the capability of really seeing what's going on with motor proteins in a cell. And I think this is a game changer for studying motor proteins. But I'm coming back at this point to the resolution question. I mean, the resolution question certainly is one that is very close to my heart, and it basically means, can we discern two molecules that are extremely close? The requirement for separating two molecules that are very close is that you modulate the emission capability. When this one is emitting, the other one should not be emitting. When this one is emitting, this one should be silent. This is the on-off capability. And this is the basic mechanism of all the super resolution methods, be it STAT, PALM, STORM, DSTORM, whatever, PAIN, SOFI. This on-off transition is key to making fluorophore separable. Now, the question is now that MinFlux really needs 100 times fewer photons. Let's imagine you have those photons. There's nothing wrong with taking more photons than you need for a certain precision. If you take more photons, if you take advantage of this factor of 100, you get a much higher precision. It's very clear. And so getting such high precisions has, of course, big benefits, because then you can imagine, say, measuring the distance between two fluorophores very, very, very precisely. Now, measuring the distance between two fluorophores is something that has been done for quite a few decades using the methodology called FRET, first resonance energy transfer. And this first resonance energy transfer means that you have a molecule that is excitable, then transfers energy to its neighbor, and then the neighbor emits, and then you can infer actually on the difference between the two. But it clearly is much better to directly measure the position of each of the molecules and then make a distance calculation, because then you get the distance, because the FRET method has its own problems with the orientation of the molecules and so on. I don't want to dwell on this. So what I'm showing you now, that MinFlux really allows you to measure distances at this scale, at this sub 10 nanometer scale. Now here the distance was prepared actually by changing the number of prolines actually between the fluorophores. So two fluorophores were attached at the end of polyproline construct, and then you have here larger distances. Of course, if you go down the ladder, you have smaller distance depending on the number of prolines. Now here the dyes are identical, so identical fluorophores, so not donor and acceptor, so identical fluorophores, and in order to make sure that they are separable, they are sequentially on and off. And they are very orthogonal in a sense that if this one emitting, so on the left-hand side, that on the right-hand side is clearly silenced because it's caged, and then if the emission stops and the first one is bleached, then the other one is activated, but the bleached one doesn't interfere actually with the emission of the second one. So we use cage compounds in order to ensure that this orthogonality works. And the high precision of minflux, the very, very high precision of minflux that we get because we need fewer fluorescent photons really allows us to measure these distances in the so-called Fred range very precisely. And here it is. So distance of 2.8 nanometers precisely measured with a precision of the order of 3 angstrom. So this is really mind-boggling if you keep in mind that this is a small fraction of small fraction of the wavelengths. Of course, it's molecular scale, and the precision is of the order of angstrom, which is even smaller than the size of the molecule itself. As a matter of fact, the minimal distance between the fluorophores is, of course, given by the size of the fluorophores themselves. It's very clear, and the fluorophore typically is 1 nanometer. So if you have two fluorophores basically touching each other, then of course the minimal distance is just given by that 1 nanometer, which is the size of the fluorophore. Now in this experiment, this was actually done. So two fluorophores were bound to protein homodimer, and then each one, of course, was activated to measure the position of this one with minflux, then this one silenced, and the other one was activated to measure the position of the other one and then silenced. And then the question was whether we would get this distance d of 1.1 nanometers, so 11 angstrom, which was predicted from structure biology. And here it is. So the distance is 10.9 angstrom, meaning 1.1 nanometer, and the precision with which we measure the distance is, in this case, below 1 angstrom. And of course, it won't get smaller than that, because, as I said, the fluorophores basically touch each other. Of course, you can get smaller numbers if you just measure projections, like here, and then it can be 6.6 angstrom or 3 angstrom, even down basically to 1 angstrom. The distance between the say average position of this molecule and the other molecule, if they are slightly tilted. The Euclidean distance, so the 3D distance between the two, must always stay about 1 nanometer. So that's very clear. This is just the projections. But it's really kind of mind-boggling because it tells you that such small distances can really be handled with lenses, focused visible light. So minflux is really capable of dealing with such small distances, and it nicely shows that sub-diffraction resolution imaging has come so far that it kind of nicely interfaces with structure biology, and this is clearly an interesting development. Now, I'm kind of shifting gears, and as I said, the decisive physical element instead palm, storm, paint, Sophie, whatever, is the on-off transition, because by on-off we make sure that the two molecules become distinct for a brief period of detection. They become separable. And this is actually the enabling element of all the super resolution techniques. As I said, without this element, none of these methods could produce an image. And I've stressed this in my talks many, many times. As a matter of fact, I even mentioned it in my Nobel banquet speech. So when I had to address the king and the queen and then the excellencies, I made the point that on-off was the key to the fact that we shared the Nobel in chemistry of 2014. And since I've said this so much, I've kind of wondered myself, maybe, God knows, maybe I'm wrong at some point, and is this really fundamentally needed? That's why I decided that we have to look into this again. I'm going to show you now that this on-off is not always needed in order to break, so to speak, the diffraction barrier and to measure distances at small scales. And why is this important? It is important because if you have an on-off process, it means that the molecule that is off can't be seen. There's no way of seeing it because, if it's off, then you don't see it. And if this one gets off, then you don't see this one. So there is an absolute need, of course, for seeing molecules simultaneously and measure their distance at sub-diffraction scale. Now, let's assume now for the moment that both molecules here are constantly on. And let's revisit Abbe's or Rayleigh's criterion again. So let's assume for the moment we have these two molecules sitting here in sample space in a confocal microscope, for example, and we scan with a normally focused beam, so a Gaussian beam, just the type of beam that you have in a confocal microscope. So here there's a detector, and here you have the beam deflectors like a Galvo or whatever, and you excite those molecules, and then you measure here the image that you get. Now the image that you would get in a confocal microscope that uses a normally focused beam is of course this one, for molecules that are close together. So these molecules are very close together, and the image of course will be kind of a Gaussian again, because the molecules are so close that there is hardly any difference to the Gaussian shape. So there's hardly any difference, as a matter of fact, to the situation where both sit in the same place. So let's assume the two now sit in the same place. They would get an image of, that just looks like this one. Yeah, there's hardly any difference. In fact, if you subtract the two, which you could, of course, then to see the difference, then you would get a kind of image or a subtraction image that looks very noisy, like this. There's hardly any difference. And so that you can make out. There's hardly, because the difference is eaten up by noise. Now, in fact, if you take a profile, if you were to take a profile in this direction, that direction, the profile of the situation where both are in the same place would be the black one, and the yellow one would be the profile of those molecules that are slightly apart. But this small difference cannot be seen really, because this is the noise band. So the gray thing is actually noise. And since noise is much larger than the difference, there's no way to separate them. That's why people said no way to separate molecules at small scales, because noise masks the difference. Okay, so this doesn't work. But let's do now a different experiment. Let's assume for the moment that our sort of confocal microscope does not use a Gaussian beam, but it uses a donut beam, okay? Use a donut beam. Now, if you have a donut beam, the donut beam has a zero intensity point of excitation light. And if you scan that donut beam across your two molecules that sit in the same place, what you will get is, of course, an image that looks like a donut, meaning that here at the center, there is no signal, because if the donut zero coincides with the position of the molecules, of the two molecules, the center of that image that you get is zero. So there is a clear zero here at the center. But what happens if the two molecules are slightly apart from each other? If they're slightly apart, there is no zero in here. There's no way of making a zero in here. Why? Because if the donut zero sits on this molecule on the left, for example, then the one on the right will emit light, and so the signal that you get at this position will not be zero. And vice versa, if the donut zero sits on the right molecule, the left one will be emitting. Or if the donut zero sits in the middle, yeah, both will be emitting. Faint emission, but there will be emission, and noticeable emission. Why? Because it can easily be contrasted with the true zero emission of the case where they sit in the same place. So if you were to do now subtraction, then you would get a signal that is really, really measurable, because you measure against zero background, you measure against zero value. And as a result, the separation now becomes measurable. You can measure them, meaning that if the molecules are very close together, you can really find out that they are not sitting in the same place. So in other words, separation becomes possible if you use a donut beam or a kind of zero intensity point of the excitation light. And it's becoming even more mind-boggling. It means if you look at the profile, then you will see that the profile that you get now is beyond the noise band. It's clearly beyond the noise band. And this is why you can separate the molecules. And in fact, the closer they get, the better this separation is. And so it's very interesting to prove this experimentally. So here you see a distance between two molecules, and this is an expected distance. How can we prepare that distance? Well, we can attach fluorophores at well-controlled distances using a piece of DNA as a ruler, so to speak. And then on the vertical axis, you can see the measurement that we got as a result. So clear measurement that shows we can go down to small values. Here, two molecules that are constantly on, not turned on and off, as you've heard before. Constantly on are still separable. So slightly less than 9 nanometers, the molecules are still separable and can be distinguished, and the distance between the two can be measured. And this is important, as I indicated already, because you can separate molecules now and watch them at the same time. So there is no need for turning on and off, and there's no need for making sure that one goes off when the other one is on, and then waiting for the right moment that the second one goes on when the other one is off. You can see them and separate them and measure them constantly. And this is, of course, critically important for tracking. Here you see two molecules that are only 15 nanometers apart, yet they can be tracked all the time and separated. And so this is really a new development, because it tells you that it is possible to, for example, attach fluorophores to a certain, say, nanoscale structure. For example, a protein. Now, protein structures are very well known by now. We know it from X-ray crystallography, we know them from NMR, we know them from cryo-EM, and there's even AlphaFold that can predict the structure. But what is less known is how they actually work. So what's the inner mechanistics of proteins? Because there's no good method that really allows you to see directly conformational changes. But now, due to the fact that we know now that we can separate molecules even if they are identical, and you don't need any on-off transition, so we don't need to silence these three when we want to measure this one, or silence the other three when we measure this one, so we can keep them on all the time. We can then watch them, all of them, at the same time and see how they change their position. And in my view, this is clearly the way to go to look into the inner mechanistics of protein conformational change, to see how proteins really work mechanistically at the nanometer scale. And as I indicated already, the physics tells us if there's zero background, the smaller the distance actually is, and the smaller change is, the easier it is to measure. And the other conclusion of this finding is that there is no fundamental need, of course, to using fluorescence, because why was the diffraction barrier broken for fluorescence microscopy in the first place? So when I looked into this problem many years back, I didn't want to break it, particularly for fluorescence microscopy, although I knew that fluorescence is important, but I was not a fluorescence person per se. However, I realized that overcoming the diffraction barrier in fluorescence microscopy is easier, because fluorophores are molecules that have different states, and you can play with the states. You can turn them on, you can turn them off, and you can put them in a certain state that has a particular feature, like emissive or not emissive, and so you can take advantage of the state transition. But once you know you don't have to make a state transition, on-off state transition, to make them separable, there is no fundamental need for fluorescence. And the reason why we use fluorescence here is the sole fact that background is low if you make a fluorescence recording. But you can imagine extending this to other types of contrast, like Raman scattering and so on, and not only to the optical domain. Principally, this applies also to X-rays, to ultrasound, or to any type of wave. Now, that's why I'm saying this transition that goes from the normal localization with a camera to min-flux is really fundamental, because this is like combustion engines. People increase the number of cylinders just to have a higher torque and get a higher acceleration, but it doesn't really scale. 2 cylinders, then 4, then 6, then 8, and 16 in this fancy car, but it doesn't really scale. And the same applies if you want to get a high precision with the normal camera localization, the square root of n, because the square root function is not a good function. It's totally different in min-flux, because if you have an electric engine in the car, there's 0 cylinders, yet you have high torque, and you can get a high acceleration. because the mechanistics actually of getting the torque and the acceleration is different. And here it is. It means that the precision scales inversely with the square root of the number of detected photons. So the more photons you have, the higher the sigma, the smaller the sigma is and the higher the precision is, but it scales exponentially. And if you ask me why is it scaling exponentially, imagine now this is a diffraction barrier, okay? And you want to know where is the molecule actually. The only thing you have to do is you have to find out, is it in this half or is it in that half? And for that, it takes one photon, yeah? So assume you find out it's in this part. And then, of course, then the next thing is, do you halve it again? Is it in this part or in that part? So you halve it and you know it's in this part. You need another photon. And then you go on with the next photon, next photon, next photon. And you can figure out that with about 20, 25 photons, you're down to basically sub-nanometer scales. Of course, in reality, it doesn't work just with 20 photons, but it shows the idea. With a few photons, you're down on the ground to nothing because it's exponentially. And this is actually the conclusion that I have. MinFlux really is a game changer. Not only allows you to get high localization precision, it also allows you to get a high spatial temporal resolution in the sense that you can localize molecules much faster, and eventually, it also allows you to separate molecules at small scales that constantly, without turning them on and off, unlike all other subdiffraction resolution imaging methods known to date. Thank you very much.
Thank you, Stefan, so much for joining us today with your incredible presentation. And if you have any questions, we have two runners that will give you a microphone to ask your questions, so be brave. And let me start, as people get their thoughts together, with the question about of the fluorophore, the chemistry of fluorophores, the half-life, the rotation, all of those other physical features of those fluorophores. Is there any progress that you see that can improve also and add to the techniques that you presented today? Half-life, yes, quantum yield, all of those.
Yes, it's a good question. Very clearly the fluorophore and the features of the fluorophore play a role. I mean, we have to separate two things now. If you look at the standard MinFlux that is already in use in many labs around the world, at least I'm aware of 50 systems. Those experiments have to be done with either switchable or photoactivatable fluorophores, fluorophores that can be turned on and off sequentially. They either blink or, what is better, they can be photoactivated. And those experiments that you've seen that are basically at small distances, like intraprotein distance measurements, they require fluorophores that are purely orthogonal in a sense that if one is active, say, emitting, the other one has to be purely inactive. Now, these fluorophores are available, and my lab has developed such fluorophores, high-quality fluorophores. Several of them actually have become commercially available from the company that I'm involved in. And so it's really critical to use the right dyes. That's very important. Now, the second version of MinFlux that I was talking about, where you use two fluorophores that need not to be turned on and off. So that's the latest thing. In that case, it's preferable to have good fluorophores that have very good quantum efficiency, that emit stably, but those do not have any requirements in terms of on-off switching. So those can be any fluorophores, any, literally any, can be any. And so I think it's a very exciting development. Still, even if you don't need that switching feature, you clearly want to have very good fluorophores. It's always advisable to acquire the best fluorophores for the experiments that you plan. So that's always advisable, because the fluorophore in the end will determine the quality of the outcome.
It is a blessing for us to have your presentation today. I am Narcisa from the UCI. I ask you a practical question from our immunohistochemistry interpretation. How far inside of a cell, 2 fluorophores have to be in order to produce colocalization. For example, in the outer segments of the photoreceptors that have a diameter of 1.4 micrometers, so 1,400 nanometers. If you have one green fluorophore on the plasma membrane, and you have a red fluorophore inside of the disc membrane, so hundreds of nanometers away, will they produce 2 separate signals, red and green, or through colocalization, they will produce yellow signal?
So the answer to your question is quite straightforward. If they are so far apart, as you were saying, more than a diffraction barrier in your case, several hundreds of nanometers, you will always see them separate as a green and a red signal. So that's not challenged by diffraction at all. And it won't appear as a yellow signal in your image. It appears as a yellow signal if they are closer than about 400 nanometers or so. Then it's starting to get yellow, and the closer they are, the more yellow it appears, although, of course, each of one is emitting green and red. If they are getting closer than 10 nanometers, what is very likely to happen is fret. So if you excite the green one, the red one will emit instead of the green one. And then, of course, your outcome is kind of scrambled by the phenomenon of fret.
Good morning. It's Audrona Lapinaite. I'm also faculty at UCI, and my lab is working on understanding molecular mechanisms of CRISPR-based systems for engineering, say, for genome editing platforms. I was very excited about your method to replace fret and observe dynamics of proteins. And I wonder what is the limitation from the time scale of this method? What type of events you can measure?
Very, very good question. You phrased it quite correctly. So what we are up to right now in my lab, and all my students, I don't have many, but I have very, very good students. We are very excited. We're very excited about this idea of replacing fret. Of course, fret will never be replaced completely because, of course, fret has its own merits and benefits. And fret is great at producing yes and no answers, very clear. But fret is not good at measuring distances because we have the 6-power dependence, as I mentioned, and we have this orientational factor, kappa square problem. The truth of the matter is that the speed, to answer your question, is limited by the fluorescent rate, nothing else, fluorescent rate. And right now, a good number to kind of keep in mind is we are right now at about precision. If you require precision of a nanometer, 1 nanometer, it takes about 5 milliseconds to get that precision. Also for colocalization, 5 milliseconds. If you're very conservative, 10 milliseconds. So anything that is slower than that will be seen for sure if it comes to dynamics. But this is not a fundamental limit. So this is really important to understand. It's with the dyes that we have used so far. So for strategic reasons, I set up a chemistry group right next door in my lab, so that I'm not really, but kind of supervising only when it comes to steering the general direction. And so one of their jobs is right now is to produce fluorophores or emitters that produce a higher fluorescent rate, and then the speed of measuring the dynamics will go up accordingly. In principle, there are papers out who claim that if you manage to group maybe 2 or 3 dyes, then perhaps you can increase the rate. I'm not sure if that works on a very small scale to some extent because there is interaction, but any measure that increases the rate of emission will increase the speed of recording accordingly. So what I kind of suspect is that if one manages to kind of get rid to some extent of the requirement to use fluorescence, and I alluded to that towards the end of my talk, then the speed would go up tremendously, and then any speed barrier would break. It would be because the rate that you get, the photon rate that you get from a laser is enormous, yeah, almost unlimited. You can have any rate, so to speak. And once we are there, then you can see the fastest possible changes, say conformational changes or whatever in a protein. So that's my dream in a way, and we hope that we get there at some point. Having said that, that wouldn't be possible without the breakthrough that you don't require the on-off. As I said towards the end, you don't fundamentally require the on-off. And this is perhaps not so well understood out there in the community that this is a major, major breakthrough. If you ask me, what is the perhaps biggest breakthrough in the last 10 years in the super-resolution field, I would definitely say this is definitely among the two perhaps most important breakthroughs in the last 10 years in the microscopy field, light microscopy field, of course, yeah. Because this is fundamental, will have a big impact.
Hi, Scott Whitcup. I'm in the drug discovery field. I have an appointment at UCLA, but your last answer leads up to my question, which is more of the process of discovery. So after years of thinking on-off was critical, what led you to challenge this? Was it data? Was it trying to solve a problem?
As I put it, I mean, I said this on-off thing repeatedly in many talks, in hundreds of talks, yeah. And to be honest with you, it was very hesitantly accepted by the super resolution community. So I was perhaps, or at least in the beginning, the only one who said on-off is really the critical element. But it was, yeah, it is really the critical element. And if you perceive it like this, and this is really critical, then of course you think, this is an Achilles heel. How can I get rid of this Achilles heel? Then the pressure, the psychological pressure on me was very high, higher than for others perhaps, because I was perhaps the only one who saw it like this as the Achilles heel. So I have to do something about this Achilles heel because it will hurt me at some point. And this is why I start thinking about it, and eventually figured out that if you do it in the min-flux way, for small fields of view, so this is important, not for big fields of view, but for small fields of view, the diffraction barrier doesn't exist. It sounds weird, yeah, but I tell you, it does not exist. If you're looking at small fields, it's okay. You can separate, yeah, you can separate any distance, and this is, it has to be point objects though. I should make this claim. It has to be point objects, and then of course the background has to be lower than the scattered signal. So this is the challenge of the matter. But hey, for fluorescence, this applies. I mean, fluorescence has extremely good background, signal to background ratio, because you have signal and background is almost zero, yeah? That's why you can see signal molecules. And of course you can have two fluorophores touching each other. And there is an important insight that I didn't say in my talk, which adds to the importance of this discovery. If you have a red and a green dye, as you've heard before, yeah, if you bring them very close, they will do FRET. They will do FRET. And if you then localize each fluorophore individually, you will localize the green one and you will localize the red one. But since they do FRET, you don't know which one actually is emitting, more or less, at some point, yeah. And this doesn't really matter. This doesn't really matter for the min-flux case. The only thing that matters in the min-flux case is there an absorption for a given point of the zero position? And it's totally irrelevant which of the dyes emit. So you can have two identical dyes, yeah, they do FRET, some homo-FRET, yeah, but it doesn't matter, because the only thing that matters is, was there an absorption for that 0 point position? And this insight is really critical, because it means that min-flux, in my view, is for fundamental physical reasons the way to go if you want to see changes in position of identical fluorophores. And I believe, yes, it has potential for drug discovery. Absolutely correct. Yes, it has, in my view. Probably it's one of the ways to look at conformational changes of proteins say, with a drug, without a drug, and hopefully, that's my dream, of course, we can perhaps reduce the number of candidates once we have understood the mechanistics of action, yeah, of the protein.
Hello, my name is Maciej Wojtkowski. I'm from International Center for Translational Eye Research, Warsaw, Poland, and I'm from imaging field. So thank you very much for your beautiful talk and also beautiful idea. But I have trouble understanding whether you can achieve isotropic accuracy in your localization with your technique. Is it possible or not? Thank you.
Yes, the answer is yes. You see, the reason why I explained everything in 2D, and that's what I did, yeah, was that I'm a person who thinks conceptually. I don't see myself so much as a developer, definitely not an engineer. Others are much better at that than me. So once I've done it in 2D, yeah, for me, it's clear that it can also be done in 3D. In fact, it has been done in 3D. So the experiments that you've seen with the polyprolines, for example, or with the dimer, yeah, the localization was done in 3D, not in 2D. So what in fact was used was a kind of, I shouldn't call it a donut. It was ring-shaped in X and Y and had some intensity above and below, okay? And so it was actually a hole. The minimum was a hole, and not a three-dimensional hole, and not just a donut. And then you get X, Y, and Z. So the localization was done in 3D, full stop. So the answer is, of course, it has to be a 3D localization, full stop. And X, Y, and Z precision are equally good. Otherwise, you could forget it. At this small scale, yeah, where you look at distances of, say, 2 nanometers, 3 nanometers, perhaps in a protein, it has to be 3D. Otherwise, it doesn't make sense, because if the protein is tilted like this and you measure only the distance, you're lost, yeah? It has to be a 3D distance. And this is done in the commercial system, yeah? The commercial MinFlux system has, by default, a 3D localization capability.
Thank you.
Hi, Stefan. This is Xiaoyu Shi from UCI. It's been 1.5 years since we last spoke at the FARM 2024. So I wanted to learn a little bit update for the deep tissue applications of MinFlux. So over the years, we've seen the success in combination of 2-photon and MinFlux, and also luckily a lot of interesting ion channels and oligomers are on the surface of cells. So your amazing dye could get over there. So seems like biologically and technologically, we are ready to see a lot of amazing applications of MinFlux in deep tissue imaging. What's your insight?
Yes. Okay. We've published a paper in PNAS quite recently where we demonstrated the use of MinFlux in biological tissue, meaning in, say, in a brain slice, yeah? And most of the work has been done in fixed brain slice, though, but also occasionally in living brain slices. Now, why does that work? Why is MinFlux suitable for that? The answer is very simple. Because in essence, a MinFlux system, if you will, is a confocal system. Yeah, it has a pinhole, yeah, no camera. Meaning that the pinhole that is here, yeah, suppresses actually the signal that is coming out of the focal plane. And because of that, you can focus the donor deeper down into brain tissue. So what we've done is we went down to about 18 micrometer. This is not much. I know some people who want to go 3/4, I don't know, they go down to a millimeter or even more. But 80 micron, that was possible, and the resolution is really very good. So it's of the order of 5 nanometers or even better. So there is, to my knowledge, no other method that can get you 5 nanometers in tissue, at least, say, 50 to 80 micrometer. Yeah. And that can be taken further. In fact, it can be improved. Why? Because we didn't use any active or adaptive optics or anything. It was just normal excitation, just normal excitation, continuous wave, nothing special, no gating, whatever, yeah? Imaging deeper in tissue, And I'm not sure whether it requires 2 photons. I'm not sure about that. I'm not saying it's the wrong way. Definitely, don't get me wrong. But I'm saying the problem with 2 photons is that the signal is usually 10 times weaker per molecule. Yeah. And you have to compensate for that. Yeah. Single photon excitation is much more benign to the fluorophore. But there's also benefits to 2 photons. I don't want to play down. Don't get me wrong. What I'm saying is not necessarily, it doesn't have to be necessarily 2 photons. So it should not be overestimated. But to cut a long story short, yes, unlike Palm and Storm, which are clearly surface bound, turf or something like that, MinFlux can go deeper into the cell, of course, and into cell layers. And as I said, we've shown it in that publication in PNAS down to 80 micrometers so far. And again, in the future, it will be better because once you apply active or adaptive optics, you can go further. And I've made a disclaimer that I've shared. That is a confocal stat and 2D MinFlux system. And that system has active and adaptive optics. I haven't tried it out for tissue, but if I were interested as a customer or a scientist in tissue imaging, I would check that system out to see how deep down I can get. Because as I said, that has active and adaptive optics, which still needs to be tried out.
Stefan, this is Michael Chang from the National Eye Institute at the NIH in the US, and I thought you gave an amazing talk. If I have a non-technical question, if that's okay. I feel like if I look at your work, what you've done is moved beyond light microscopy, and which is, I assume, a 19th century technology, into these amazing 21st, even 22nd century technologies of trying to develop methodologies to improve the resolving power, what you can do to visualize. For ophthalmologists, as eye doctors, in some way, eye doctors are doing the reverse problem, that eye doctors are trying to measure the resolving power of the eye. And the way that they do it is the equivalent of that 19th century methodology is you give a patient an eye chart, and what's the smallest letter you can read. And that's a measure of what the resolving power of the eye is. Have you thought about, for eye doctors, are there 21st century methods that we could be doing better to measure the resolving power of the eye?
It's a very good question. And actually, I haven't thought about it that way. I will start thinking about it. No, the answer is no. I haven't thought about it because I was focused on my problem with microscopy, but I haven't thought about it from the perspective of an of an eye doctor. But it's a good point. Maybe, it's very obvious that the eye is just something, and there is also the brain that processes what you see. So I'm not so sure to which extent you can also take that into account with something that is more objective. But the objective part that is independent from the brain, perhaps, could be done better. But I have not thought about it.
All right, thank you so much. Please join me to thank Stefan for a really inspiring presentation. And Stefan, thank you also for the inspiration how to do presentation by the Zoom. That was something that I have to do in the future if it comes to that. Thank you very much.